Let's assume a school. There are several things going on there, but everything is controlled by the office of the headmaster, and that office in the case of a cell is known as the nucleus. A cell has to do its metabolism as well as its functions in a regulated manner, and that is controlled by the genetic material which is present inside the nucleus. The nucleus is a membrane-bound organelle that is present inside the eukaryotes. In the most cases, single nuclei is present inside a cell. But there are some exceptions as well. In the case of a mammalian RBC, no nucleus is present at all, while in the case of some mussels as well as in a fungal hyphae, there are multiple nuclei present inside a single cell, and that condition is known as a syncytium. The nucleus is having the protection of two lipid bilayer membranes, and out of which the outer membrane continues to form the endoplasmic reticulum. Inside the nuclear membrane, we can see the nuclear matrix, which actually includes the nuclear lamina. The nuclear lamina is mostly made of intermediate filaments and a protein which is known as laminin. The nuclear lamina is involved in providing support as well as several other functions like chromatin rearrangement, cell division as well as DNA replication. And like a school, there is a rule book for a cell, and that rule book in the case of a cell is known as the genetic material. Most of the genetic material of a cell is actually found inside the nucleus except for some extra chromosomal DNA like in case of mitochondria as well as chloroplast. According to several literatures, this microscopic cell in my body is having a DNA of like 3 meters long. And this 3 meters long DNA is actually packed inside this cell. And one of the major role in this condensation is done by a protein known as histones. The histone protein actually contains two basic amino acids, lysine and arginine. Due to these amino acids, the histone is having a positive charge and the DNA is having a negative charge. Due to this positive and negative interaction, the histone can effectively bind the DNA and condense it down. And after the condensation by histones, several other factors like non-histone proteins as well as the nuclear laminins also involved in condensing it more. The human cell is having 23 pair of chromosomes, and it is perfectly arranged as these chromosomes only at the time of the cell division. And in the normal metabolic times, they are loosely wound and they form the chromatin. And there are two types of chromatin as well, the euchromatin and the heterochromatin. The euchromatin region of the DNA is actually lightly stained, and it is the region where the expressing genes are present, while the heterochromatin is actually the darkly stained area, and it is the area where non-expressing genes are present. And there's the dark, discrete body present inside the nucleus, and it is known as the nucleolus. The nucleolus is the ribosome factory where ribosomes are produced, and it is involved in the production of rRNA as well as the ribosome assembly. Snapshot time. In this video, we have studied about the structure of nucleus and the genetic material. Also, we have studied about the nuclear laminin and the nucleolus. The ribosomes are actually produced inside the nucleus, and they needs to be transported out of the nucleus for their functions. And do you know how it is transported? It is actually done by the nuclear pore complex. Do you know what is a nuclear pore complex? And that's going to be the topic of your next video. That was your 3 minutes. See you in the next one.